This is Dr. Mobeen Sayed, and today we are going to talk about the loading of MHC II complex or major histocompatibility complex number 2 and how it is loaded with an exogenous antigen. So let's start our discussion. So MHC II loading, that is the objective for the discussion today. We're not going to talk about MHC II and 1 comparison, but really the mechanics of loading MHC II. So MHC II or HLA class 1, 2, and 3, MHC II is the class 2, they can be used interchangeably. So HLA stands for human leukocyte antigen. These genes that encode them, they are present on the chromosome number 6 on the petite arm or the small arm, and this is the area where they are present. In this area, the genes that are present are basically class 1, 2, and 3. And class 3 is actually interspersed between 1 and 2. Here, I'm only showing the class 1 genes and class 2 genes. And an easy way to remember the genes is that the one-letter genes are for class 1. So HLA B, HLA C, HLA A are all for class 1 MHC complex. And the two-letter genes are for HLA2 or MHC 2. So DP, DQ, DR are all two-letter and they make HLA class 2 complexes. Another thing to remember is that for class 2 that we are discussing today, for class 2 there is a beta chain and there is an alpha chain, and they both are encoded near each other. So beta and alpha chain. On the other hand, for class 1, the alpha chains are encoded on these genes, and then beta chain is separately encoded on a different gene, on a different chromosome. So to start our discussion for loading, this is for the exogenous loading. That means the pathogens or environmental agents that are present around the cell will pick that up and then present that on MHC II. So here is a cell, some structures that we should keep in mind when we are looking at the loading. The right next to the nucleus, this is the nucleus. Right next to the nucleus is rough endoplasmic reticulum. Rough endoplasmic reticulum is actually connected with the nucleopores so that the messenger RNA that comes out of the nucleus directly enters the endoplasmic reticulum. The endoplasmic reticulum in turn is studded by, it is attached by many ribosomes, which will then look at that mRNA and create proteins. Then next to the rough endoplasmic reticulum is the Golgi apparatus. Golgi apparatus sits near the rough endoplasmic reticulum so that the proteins that eject from the rough endoplasmic reticulum, they can go to Golgi apparatus for further processing, packaging, and secretion if needed. So with this, let's quickly look at the structure for a second of the MHC II complex, and then we'll see how it is loaded. So the MHC II complex is made up of two chains. So here, if you see, this is the alpha chain, and it has two parts, alpha 1 and alpha 2. And the second part has a transmembranous part as well, which will be anchored in the cell membrane. Similarly, beta chain also has two parts, beta 1 and beta 2, and that also has a transmembranous part that will be anchored in the cell membrane. When the MHC II complex is created, this area here, where you see this green area, molecule present, this area is actually the binding site. This is where the antigen will bind. However, we do not want the MHC II to start binding with self-antigens. So we pack it with another protein which is called invariant chain. And if you see, invariant chain is all of this little red molecule over here. The part of the invariant chain that passes through the binding region is called clip. So we'll discuss that in a second as well. So you can think of this as an MHC II complex that is packaged by a packing which prevents it from biting the self-antigens. So now let's look at how the loading will occur. So here is the nucleus. the nucleus will make messenger RNA for making MHC II protein. So within the rough endoplasmic reticulum, the ribosomes that are attached here, they will look at this messenger RNA and they will manufacture alpha and beta chains that will be then assembled together, and then an invariant chain will be attached to them as well. So kind of creating a pack. Usually this MHC II complex is trimerized with other two more complexes as well, but that is not important at our level. So then what happens is, from the rough endoplasmic reticulum, this MHC II complex will be packaged into a small vesicle and it will be transported to the nearby Golgi apparatus. It is the cis phase of the Golgi apparatus where this package will be coming in and it will fuse with the Golgi apparatus. So once it is fused here, it is now for further processing in the Golgi apparatus. With the further processing, what will happen now is, if you keep an eye here, here is the processed MHC II complex. This MHC II complex will be then pinching off of the Golgi apparatus and will start traveling towards the endocytic vesicles. So now this is a very fascinating area. So hold this thought for a second and let's go to the cell membrane. So imagine this is the outside of the cell and there are various kind of environmental factors there. Maybe there are some pathogens present as well, some viruses present, fungi. And what cell is doing is continuously, the cell is taking a sample of the environment and bringing it in. If the sample size is bigger, then that will be called a phagosome. If the sample size is smaller, then that is called endosome, or endocytosis is the process. Pinocytosis also means a smaller piece that is picked up. Generally in the literature, when you would read endocytic vesicle, that would actually mean both kinds, phagocytic or pinocytic vesicles. So here we have a bacteria that got trapped into this endocytic vesicle and it is brought in. Now endocytic vesicles, when they are brought in, they go through three stages: early, late, and then fused. So in the early stages, the acidity within the vesicle is lesser. For example, I think it is near 6. Then it falls to 5.5. The vesicle starts becoming acidified. Those acidic enzymes that are present here, they would start breaking down the pathogen or whatever sample from the environment that has been picked up. However, it will be the late vesicle, endocytic vesicle, that would fuse with the lysosomal compartment, and the lysosomal enzymes would further come in, further reduce the pH to 4.5 or near that, and start digesting and breaking down the pathogen into smaller antigens. So this is happening on a continuous basis. On the other hand, from this side, the MHC II complexes sitting in vesicles are also produced on a continuous basis. When the MHC II molecule and the compartment, molecules and the compartment they are sitting in, when they come out of the Golgi apparatus, the tail of the MHC II, so let's go back for a second to the MHC II complex here. This tail of the MHC II would actually be sticking out of the vesicle. So if this was a vesicle around this, the tail is sticking out. This tail, the part that sticks out of the vesicle, would allow the trafficking proteins like dyneins and kinesin to pull this vesicle and fuse it with the late endocytic vesicles. Isn't that fascinating? So the tail has sort of a visa on it or a shipping label on it to say, please take me to an endocytic vesicle. So that is how the MHC II complex will be fused with the late endocytic vesicle, which is also fused with the lysosomal compartment. So digestion of the antigen is happening, the pathogen is happening, and the binding proteins, which are the MHC II complexes, have arrived in that combined compartment as well. So if this is that compartment, what you're seeing is that this is the MHC II complex. It would ideally, this should be anchored over here and the tail should be sticking out. Then this complex is here as well. And what happens is various proteases start acting on this invariant chain and start cleaving it away. So in the first go, if you see here in this diagram, they would remove this part of the chain. Then in the second go, they will remove this part as well, and we would be left with an MHC II and a clip region that is still blocking the binding region. Finally, this clip protein will also be removed, and the antigen will be loaded in that area. So imagine this little antigen is part of the bacteria. Once that happens, just to quickly review that, this was the MHC II, this was the invariant chain, alpha, beta chains, membrane anchoring parts, the invariant chain with the clip region, then the first cleavage occurs and part of the invariant chain is removed, clip region is there and the tail is there, then the tail is removed as well and the clip region is left, and finally in that clip region, when that is also removed, instead of saying clip region, the clip part of the protein, when that is removed as well, the binding region becomes available where then an antigen is bound. Now what happens is, if you see here, once this antigen is bound, as we saw before, then this piece or this small part will be pinched off of this compartment, and that will become a vesicle. This vesicle will be trafficked to the cell membrane, where when this fuses with the cell membrane, automatically then the MHC II with the antigen bound to it becomes expressed on the surface. So this is how the MHC II is loaded onto a cell surface. And this is a continuous process. The cells are continuously sampling the environment. So the endocytic vesicles are continuously coming in. They are continuously fusing with the lysosomal compartments. From the Golgi apparatus, continuous supply of the MHC II packaged into vesicles, sitting on the dynein and kinesin proteins on microtubules, running towards those compartments. And they would go, all three of these things will fuse: endocytic vesicle, lysosomal compartment, and the MHC. And then the invariant chain will be removed and the binding region will become exposed, and over there the antigen will be bound, and then all of that package is sent to the cell surface, and that is how MHC II molecule is loaded with exogenous proteins. Now remember this, that in some cases the macrophages and dendritic cell can actually have endogenous proteins presented on MHC I and II together as well without getting infected. You would see in the MHC I lecture that the endogenous proteins that come into a cell, for example, a virus that infects them, will be presented on MHC I. But there are some cells that can present antigens on MHC I and II simultaneously without getting infected. So that is a discussion for today. I'll see you for the MHC II soon.